Late into the night, biochemist Brigit Liss works in her Oxford laboratory, probing secrets held deep in the minutiae of single brain cells. She dreams of unlocking the mysteries of a distressing illness. She wants to unpick the puzzle that is Parkinson's disease.
Parkinson's is a really common disease, and actually the cause is still unknown. But what we know is that specific nerve cells in the brain die in Parkinson's, and that makes all the symptoms that you have problems with voluntary movement. If you say, I would like to grab this mug, I'd simply do it. A Parkinson's person had problems to do it, to initiate the movement. And so we know that the symptoms come from the degeneration, from the dying of the specific group of nerve cells in the brain, the so-called dopaminergic nerve cells, dopaminergic neurons. And in Parkinson's, many of them die in the course of the disease, but some manage to survive. So we are interested in finding out why can some of them survive their assault and some simply die. And so there must be a difference in the function that enables them to survive.
And it's that difference in function that is the focus of Brigit's research. Working with delicate precision in her lab, she studies the minute building blocks of life. There are hundreds of types of brain cell. The ones that are affected by Parkinson's are known as dopaminergic neurons. In order to find out why the disease causes some of these particular neurons to die while others survive, Brigit must isolate individual cells from a sample of mouse brain.
I'm looking for a nice neuron that looks healthy and fine. This is a neuron, and here's the tip of my pipette.
Having honed in on a single cell under the microscope, Brigit maneuvers her tiny glass pipette against the membrane. Amazingly, her equipment can read the cell activity and identify it as a dopaminergic neuron.
They are most beautiful neurons, but this is of course because I work with them, and other scientists have other neurons where they work for, and they said, no, mine are much more important than yours. But I can always say mine are important for voluntary movement, they are affected in drug addiction, they are targets in schizophrenia, they are targets in Parkinson's disease. These dopaminergic guys are also important in another brain region called the hypothalamus. For example, they are important for eating and modulating a lot of functions. So I always say they are important for everything which we like. They are involved in sex, for example. They are also in pleasure, everything. So I think there's a lot of we know about them, but there's a lot of things we don't know. And as long as we don't know each other, if we don't understand them properly, we can't understand what's going on when somebody's ill, when you get Parkinson's, when you get schizophrenia, when you get drug addicted. As long as you don't know how they function in health and control condition, you will never understand what's going on when something's going wrong, when you get ill.
Brigit's passion for biochemistry is shared by her friend and mentor, Professor of Physiology Francis Ashcroft. As a fellow scientist, Fran understands the skill of what Brigit does and why it matters.
The reason it's very important is because in your brain, the nerve cells that sit adjacent to one another are not identical. So you can have one cell doing one thing and another cell doing another thing, and that's quite different from what you find in the muscles of your arm, where all of them are the same and doing the same thing. So you have to be able to look at single cells if you want to know how the brain works. And that is extremely difficult. She's one of the few people in the world who are getting this to work. So that's very novel.
Brigit needs to work at this single cell level because she wants to study the differences in the genes that are active in individual neurons. Although every cell carries a copy of every human gene in its nucleus, it uses only some genes at any one time. When a gene is switched on, a copy of the gene is made in the nucleus and then sent out into the cytoplasm of the cell. By extracting and identifying these copies, Birgit hopes to find out exactly which genes are switched on in individual neurons. The first job is to extract the cytoplasm from within the cell.
That was a nuclear? Yeah. Haha. Now we can harvest the rest. It's no easy.
Harvesting cytoplasm may be easy for Birgit, but her work impresses other people. The Royal Society thought enough of it last October to award her a fellowship and an accompanying grant. New College Oxford have also awarded her a fellowship. As part of that, they allow her, rent-free, the use of one of the college-owned flats in North Oxford. This is a place of her own, where Birgit can escape the pressures of the laboratory. There was never any doubt about the path Birgit's career would follow. Even at a young age, she was drawn to biology.
We had a kind of medical dictionary at home. And when I was 6, I always grabbed this dictionary and read it from A to Z. Because, oh, it's so fascinating, it's so great. And my mother was crazy. And she went then to school, to the teacher of my brother, saying, I have this little girl there, and she's reading this medical dictionary. And they said, oh, that's unusual, but leave her with that.
In the scientific world, nothing stands still for long. Although deep in research, Birgit has moved laboratories.
At the moment, actually, it's quite exciting because a lot of things have changed, and point in career where I'm for the first time completely in charge of everything concerning my research.
Birgit's various grants give her financial independence, so she has free rein to decide what she researches and with whom. The flip side of this freedom is the admin, applying for grants, running a budget and supervising staff.
The more advanced you are, the more you come into having your own lab, your own group, their own people under you, the more you just sit on your office. Actually, most of the professors, they never see the lab anymore. Of course, they have a look inside, but they never do any experiments anymore. But I very much hope that I will always do experiments because I enjoy it so much. But
part of Birgit's role involves teaching. She's instructing her PhD student in a procedure that is vital for her own experimental work.
Yes, the time 1 minute at 72 degrees.
Called PCR, the technique allows a scientist to make many copies from just a tiny amount of genetic material.
I started establishing this technique in Germany and during my PhD thesis, and have over the years become more and more routine in it. But then, of course, you adapt it to your own needs, to your own experiments.
PCR has a wide range of applications. Controversially, forensic scientists use it to amplify DNA found at a scene of crime. In Birgit's research, it is used to multiply the information she has so carefully collected from single dopaminergic brain cells. From one copy of a gene, the technique can make millions more. Birgit will need a lot of such material to test her theory that reaction to the effects of Parkinson's disease might be related to gene differences. Birgit's home is near Hamburg in northern Germany. Modern science is an international field and travelling becomes part of the job. She's been in England for 3 years now. In career terms, it's been a good move.
Which goes, over days. Bye, guys.
To come to Oxford, she's had to leave her country behind for the sake of science. Visits from old friends are a welcome reminder of home.
She feels quite comfortable, but I guess sometimes her friends are missing. And I guess she misses the German food. That's why I always bring a sort of survival pack. Suitcase full of German food.
If you want to be a scientist and your personality is that you look for security, it's not really possibly the right job. And because you never have, for example, after your PhD, you don't get simply a job, a permanent position or whatever. It's not like that. You get maybe a 1-year contract as a postdoc or a 2-year contract at the next postdoc at the next place. And you have to change your lab 1 or 2 or 3 times in order to pursue your career. That's normal. So you for example, if you plan to have a family and children, it's not that easy because you have no long-term perspective in first instance.
The next stop on Brigit's Parkinson's quest takes her to a laboratory close to Oxford. Any one of the thousands of genes in human DNA could be involved in Parkinson's disease. Scientists here have developed a way of multiplying material from cell cytoplasm so that all active genes are amplified equally. This is a revolutionary step forward from the PCR method that Brigit can do alone, and it means the full range of genes can be studied at the same time. To identify the active genes, Brigit will use a technique called microarray. The microarray has transformed genetic testing because for the first time it lets you test for thousands of genes simultaneously. Commercially produced slides are coated with copies of all the genes that might be active. Brigit will mark the genetic material she has harvested with fluorescent dye so she can see where it goes. When the fluorescent sample is washed over the microarray, individual genes will stick only where there is a matching sequence on the slide. And when the microarray slide is analysed, active genes present in the sample will show up as fluorescent spots. This could tell Brigit which genes are active in the brain cells that die from Parkinson's and which are active in those that live.
I get it out here, yeah?
Working with Harwell chemist Claire Pritchard, Brigit prepares the experiment.
Is there right orientation?
That's the right way up.
Mm-hmm.
The big question is whether the precious amplified material will produce intelligible results. Brigit has never performed a microarray test before, and one of this type is also new to Claire.
It's completely new for me and really exciting. This is a novel thing which we are quite excited because neither me nor you, we have done it so far.
A scan of the array slide will tell them whether their work has succeeded. Positive results will open the door to a whole new area of research for Brigit.
Explain today, possibly, yeah? So what we get is a kind of so-called false color picture. The computer gives it a color that we can see with our eyes, but of course the fluorescent light normally we can't see. So the computer is making colors up for us. Yeah, but it has worked. We get a signal, and that was what we had explained. The bottleneck was to get enough material that you see something on the array, which we obviously have.
So the results are encouraging. Unpicking the puzzle of Parkinson's is still a long way off, but Brigit is not about to give up.
Of course, my main aim would be to really find something out that makes a difference. Possibly in terms of Parkinson's disease, a little bit possibly. That would be my dream.